Human skin, the body's largest organ averaging 1.5 to 2 square meters in adults, is a dynamic and complex organ, far from passive, constantly interacting with its environment through thermoregulation, DNA protection, and immune support. It regulates temperature via up to 4 million sweat glands per person, protects our DNA from UV-induced thymine dimers, supports innate immunity through antimicrobial peptides like defensins, and crucially, responds to sunlight wavelengths for vitamin D production. The diverse colors of human skin, determined by melanin brown-black and pheomelanin red-yellow pigments, are a profound reflection of deep biological adaptation to varying environmental pressures over evolutionary time. Modern humans first evolved in equatorial Africa approximately 300,000 years ago, where intense sunlight with UV index often exceeding 12 year-round was a constant environmental factor shaping survival. This high ultraviolet radiation, up to 50 kilojoules per square meter per day at the equator, posed continuous threats by penetrating to the dermis and damaging cellular DNA through cyclobutane pyrimidine dimers at rates 10 to 100 times higher than in temperate zones, while also degrading essential nutrients like folate. Within the epidermis, specialized cells called melanocytes, numbering about 1,000 per square millimeter or 1 per 10 keratinocytes, synthesize melanin using the enzyme tyrosinase. This melanin is packaged into melanosomes 0.5 to 1 micrometer in diameter and then distributed via dendrites to surrounding keratinocytes, forming protective supranuclear caps over the nuclei. Melanin effectively absorbs 50 to 75% of UVB radiation 280 to 315 nanometers and 99% of UVC below 280 nanometers, significantly limiting DNA damage, quenching reactive oxygen species like superoxide, and reducing penetration by up to 90% compared to lighter skin. Ultraviolet radiation at wavelengths 290 to 330 nanometers also degrades folate in the bloodstream, specifically photolyzing 5,10-methylene tetrahydrofolate to inactive products, a vital nutrient essential for cellular processes. Folate is essential for DNA synthesis and methylation, spermatogenesis, where deficiency drops sperm count by 20 to 30%, fertility, and healthy embryonic development, including neural tube closure, where risk of spina bifida increases tenfold in deficiency. Dark pigmentation preserved folate levels by blocking UV degradation, which directly increased reproductive success in high UV environments, as shown in mouse models where UV-induced folate depletion have litter sizes. In these high UV regions like sub-Saharan Africa, individuals with higher melanin levels were more likely to reproduce successfully due to better protection against DNA damage and folate loss. Over many generations, with selection coefficients estimated at 0.1 to 0.3, this natural selection led to dark skin becoming the dominant trait. Fixed high melanin alleles at loci like MC1R, potentially within 10,000 years, with evidence from fossils suggesting dark skin by 1.2 million years ago in Homo erectus. As humans migrated northward and eastward out of Africa around 60,000 to 100,000 years ago, reaching Europe about 45,000 years ago, and East Asia 40,000 years ago, sunlight intensity and UVB flux significantly declined. In these new environments at latitudes like 45 degrees north where UVB drops 90% in winter to less than 0.5 minimal erythemal dose per day, high melanin levels, while protective against excess UV, reduced the penetration of beneficial ultraviolet radiation needed for other functions. This created a new evolutionary constraint, the need for vitamin D synthesis in the skin to prevent deficiencies leading to conditions like rickets. Vitamin D production in the skin, converting 7-dehydrocholesterol to pre-vitamin D3, is directly dependent on exposure to specific ultraviolet B wavelengths of 295 to 315 nanometers, with peak quantum yield at 297 nanometers primarily in the stratum spinosum keratinocytes. In low UV regions, excessive pigmentation limited this crucial vitamin D production, requiring dark skin 5 to 10 times more UV exposure to achieve target serum levels of 20 to 40 nanograms per milliliter. Consequently, genetic variants that reduced melanin production, such as those in SLC24A5 and SLC45A2, became advantageous, leading to lighter skin tones through natural selection. Diet also modified selection pressures. For example, populations with vitamin D-rich diets from fish providing 10 to 20 micrograms per day, like the Inuit or Sardinians, could retain darker skin even at higher latitudes. Furthermore, the development of clothing around 40,000 years ago with needles by 27,000 years ago in shelter, along with agriculture 10,000 years ago reducing mobility, altered sunlight exposure by blocking up to 95% of skin surface. Beyond UV protection and vitamin D synthesis, skin covering 1.5 to 2 square meters is a vital immune organ with barrier lipids like ceramides comprising 20 to 30% of its composition. Pigmentation influences inflammation by modulating TLR4 signaling and reducing NF-kappa B activity, microbial defense, and the stability of the skin barrier, with lighter skin showing higher risk for conditions like psoriasis. Local pathogen environments, such as tropical diseases like malaria, also played a role in shaping optimal pigmentation levels through interactions with skin barrier lipids and immune signaling. Skin color evolution is polygenic, involving over 100 genes such as SLC24A5 with its A111T allele explaining 25 to 38% of variance in Europeans, rather than a single pathway. Different populations developed light skin independently through convergent evolution, for instance SLC45A2 in Europeans and East Asians, and OCA2/HERC2 variants around 11,000 years ago. Migration bottlenecks, such as those with around 1,000 founders for Europe, also amplified certain traits by chance through genetic drift, contributing to the diversity we see today. Human skin color exists as a continuous spectrum with global reflectance varying from 20 to 70%, showing clinal variation of 1 to 2% per degree of latitude, not discrete categories. It reflects adaptation to environmental UV gradients, climate, nutrition, and pathogens, not hierarchy or social constructs, as a classic example of natural selection. Evolution optimized survival and reproduction through balanced pigmentation, protecting against UV damage while enabling vitamin D synthesis, nothing more, nothing less.